Thank you very much to the organizers, especially Dr. Anuradha, for giving me this opportunity. And my lab here at NCBS is interested in understanding the epigenetic mechanisms of cancer inheritance. And what I mean by that is that we are particularly interested in, sorry, my slides are frozen. Okay. So we are particularly interested in understanding the missing heritability of traits such as cancer. So what I mean by missing heritability is that so far the mutations in coding genes that we have been hearing in this meeting can explain the 30% of cancer heritability. However, there are families where cancer runs through generation, but if you do the NGS, probably you don't find mutations in the coding part. And this is where we hypothesize that this missing 70% heritability of cancer can be explained if we understand the mutations in the non-coding genome. And why I say that understanding non-coding genome is important is because if we take the mutations that are associated with cancer, or for that matter in any other trait that are identified by genome-wide association studies, we see that majority of these mutations are present in intergenic sequences, and only a few mutations are present in coding genome. Now, if a coding genome part, such as the gene, is mutated, we understand that protein can be truncated, destabilized, or enzymatic activity can be affected. But we don't know if the intergenic region, that is non-coding genome, if there is a mutation, how that can give rise to cancer. So what is there in the non-coding genome? We know that the coding part, there are only 30,000 genes, and it is only about 1.5 to 2% of the genome. And this coding gene have promoter, exon, introns. But apart from that, the large swaths of genomic regions that are non-coding genome has some regulatory elements that are known as enhancers. And by genomic approaches such as ChIP-seq, we can identify where the enhancers are in different type of tumors. For example, lung tumor will have different enhancers, whereas the breast tumor would have a different enhancer. And how we identify them is by ChIP-seq for such histone marks like K4-dimethyl, monomethyl, or K27-acetyl. So what these enhancers do is they give a meaning to the cell. So for example, these are the bunch of stem cells that are completely identical in terms of their DNA, genomic DNA. But these stem cells, upon differentiation Q, they can give rise to different type of cell types who will have a completely different function. And this function is because the genes that are there in the stem cells are differentially expressed. And this is where enhancers are important. Enhancers decide which promoter would be activated. Enhancers are the key driver of genome expression. And what they do is, once enhancers, they bind with protein machineries such as transcription factors and polymerases, they produce these non-coding RNA which are known as eRNA. Then enhancer finds its target promoter by bending the intervening DNA, and this is called looping. So this is how enhancer then activates the promoter. And we know that certain enhancers in, for example, in breast tumor get hyperactivated, such as they activate the MYC oncogene or the other oncogenes. And this is how these genes, oncogenes, are hyperactivated. It's just because of enhancers. So today I'll give you an example how mutations in enhancers in non-coding genome can heighten the susceptibility for a tumor. So during my postdoc, I and a colleague had found that majority of the Jiva's functional mutations were present in enhancers, and the same concept we utilized in my lab now, and we have found the enhancer mechanism that would probably explain the genetic inheritance of disease. So today I'll give you example of one of the example that we have shown, that how enhancer mutations can give prostate cancer susceptibility to men of African ancestry, and this is one of the example, and the same method can be used to understand this such heritability in other tumors also. So we know, for example, the men of African ancestry have about 3-fold high chances of having prostate tumor over the other ancestries. And if you look at the jivas done on the prostate tumor, again we see that most of these mutations that are being identified, again they are in the non-coding genome. So again, I told you that the non-coding genome has enhancers. So our hypothesis was that maybe these mutations are affecting enhancers. So towards that, one of my collaborator, Chris Heyman, in University of Southern California, he performed Jiva studies in 10,000 controls and 10,000 prostate cancer individuals from the African ancestry. And he found that there was a hotspot SNP, is called 72725854, which is present in the famous 8q24 locus. So if you look at 8q24 locus, there's only one coding gene, MYC, but neighboring genes are all non-coding genes, and they produce long non-coding RNAs. And the SNP that he identified was between these two long non-coding genes, PCAT1, PRNCR1, and PCAT2. And we know, for example, that if PCAT1, which is long non-coding RNA, though it is not coding gene, when it is hyperactivated, it allows cells to grow rapidly, divide fast, and when cells divide fast, they don't have time to repair their DNA, and that is where cellular transformation happens and the cell becomes cancerous. So overexpression of link RNA at this locus is associated with tumors. So anyway, so this enhancer, sorry, the SNP that we identified, we asked whether the SNP is present in the enhancer. So we overlaid all the epigenetic marks that are present in the prostate cancer cell, and we identified indeed the SNP that we have found in African ancestry happens to be present on the enhancer in the non-coding genome. And as we can see, this mark of K27 acetyl, Pol2, these are all marks of active regulatory regions such as enhancer. So what this enhancer is doing? Towards that, first we understood if this enhancer is really functional. So we cloned this enhancer in a luciferase reporter assay, and we see that this region which has the SNP is actually an enhancer. Then what happens to this enhancer when the risk allele T is present, whereas if the non-risk allele A and G is present? This is a triallelic SNP. So what we see is that when T allele is present in the enhancer activity is much more robust. So what it suggests is that individuals who have this particular SNP, the enhancer becomes hyperactivated. So how it becomes hyperactivated? We know that the prostate cancer is driven by androgens and testosterones. And once testosterone is present in a cell, there is a transcription factor called androgen receptor that goes into the nucleus, binds with DNA, and activates the genes. So we ask if the enhancer is responsive to testosterones in the men of African ancestry. And we are surprised to see that when cells were given the testosterones, the non-risk SNPs A and G were non-responsive. However, the T allele, which is a risk allele, when it was present in an enhancer, the enhancer became even more hyperactivated. And also it suggests that when T allele is present, the enhancer now responds to higher levels of testosterone. And this might be important after attaining the puberty when the testosterone levels are high. Maybe in the individuals who carry this T allele, the enhancer becomes hyperactivated. So as I said that enhancers control the promoters. Now we know that this is the enhancer where the mutation exists. What genes it is controlling? So towards that, we see that this is the enhancer here, and there are many genes around it. So to understand which genes are regulated by this enhancer, we performed another experiment. It's called chromatin conformation capture, which is nothing but it identifies the genomic interactions between different regulatory elements in the genome. And when we did this experiment, we found that indeed enhancer is interacting with PCAT1, PCAT2, and PRNCR1. These are all long non-coding RNAs. So we blocked the enhancer to validate if these genes are regulated, both PCAT1, PRNCR1, and PCAT2. And what we found was that when enhancer was inactivated or blocked, the expression of all long non-coding RNA, including PVT1 and the coding gene MYC, was downregulated, suggesting that enhancer is regulating these long non-coding RNAs and MYC at 8q24 locus. So with help of Sabri, we found that in the TCGA data sets, the in the pan-cancer datasets, the tumors that had heterozygous risk allele or the homozygous risk allele, the expression of these long non-coding RNA in MYC was high, again confirming our hypothesis that this enhancer, when there is a T allele, the expression of this link RNA in MYC is high. And also, even in the prostate adenocarcinoma, we saw that when T allele was present, the expression of this link RNA in MYC was high. Then we identified that when PVT1 expressions were high, the survival rate of these patients was really poor, and also the alterations in MYC also reduced the survival. Interestingly, we see that when PVT1, which is a long non-coding RNA, when its expression is high, the Gleason score of tumors was also high, suggesting the tumors are much more aggressive, which is true because men of African ancestry, when they come to clinics, often their Gleason scores are much higher. So what is the mechanism? Towards that, we see that when T allele is present at the enhancer, it gains the binding site for SPDF. SPDF is a transcription factor, and through tumor analysis, we observed that SPDF expression is much higher in tumors, and it's highly expressing in prostate tumors. So we know that this protein is relevant to prostate cancer, and when individuals have T allele, the T allele binds with SPDF, which we confirm by chip experiments that T allele binds with SPDF, and when we knock down SPDF, the enhancer is no more responsive to testosterones. So to summarize, what I've shown you today is that individuals who have non-risk allele, enhancer is there, very much there, but it is dormant. However, when A is mutated to T, a germline mutation, this T causes the binding site for a transcription factor SPDF. Then SPDF recruits androgen receptors, cofactors, the data I didn't show you in interest of time. So this enhancer becomes hyperactivated, and once it is hyperactivated, it starts to loop with neighboring long non-coding RNA, and these long non-coding RNAs are already known to play an oncogenic role in various types of tumors, including prostate tumors. So this is how we show that probably in these individuals who carry the risk SNP, when they attain the puberty, testosterone levels become high. That is when this enhancer becomes hyperactivated, and that is when these link RNAs are highly produced. Their expression increases, and these are the individuals who are now prone to prostate tumors. So with this, I would like to thank my collaborators, Chris, Matthew, and Sabri, and also the funding agency and my students. And I'll be happy to take any questions in later discussion. Thank you.
Thank you, Dr. Dimple, for very nice introduction regarding epigenetic mechanisms in cancer. Now we will proceed with our molecular tumor board discussion, and we will request all the speakers to kindly answer questions in the chat box. Because of time constraint, we will not be able to discuss them over here. So our next molecular tumor board will be moderated by Dr. Smruti, who is oncologist at Bombay Hospital. Requesting Dr. Smruti to kindly invite all the panelists and proceed with the discussion. Thank you.
So Pratik, there is no time for questions. I mean, these were very interesting talks.
Yes, sure, sir.
Is there time, or is there
we can cover one question?
Okay, I mean, maybe one question. I mean, both of these people, they are the stalwarts. I mean, the upcoming cancer genomics from like India. Very happy to see them at PMH. Thank you both of you for coming over here and presenting. So maybe my first question to Sabri. Sabri, was this kind of a bit of a surprise and a bit of a bummer actually, that the whole purpose of doing the whole genome sequencing is to look for something that what Dimple was focusing on, something even beyond the coding regions. But given that majority of the mutations or most of the mutations that what you found were the driver alterations alterations that I'm primarily addressing too, that you were finding were in the coding regions. So how do you account for? I mean, was that this statistical issue? You were not powered enough to find driver alterations, which is outside in the coding region, or there were some other reasons that underlies it?
Yeah, thank you. It was a very interesting question. Right now, I don't have a clear answer to this because, as we know, that some of the coding regions, because of its functionality, all the driver events happening at the same time. So the recurrent event happens within the coding, whatever the domain it is. But when we go to the regulatory landscape, as Dimple has mentioned, the enhancer regions are quite big. So also, if you have a super enhancer, you have a stretched of enhancers too. So the current statistical approach where you try to look for recurrent in a short region may not capture those kind of scattered driver events. So I think both in terms of genomic annotation for the proper regulatory region and the statistical approach may be one of the reasons that limits the identification of drivers.
But looking ahead, you do anticipate driver alterations as what Dimple has presented.
Yes,
even that may have a significant role to play this.
So we need to incorporate more chromatin and regulatory information, which Dimple suggested to take into account when we detect for this driver event. I think there's a way forward. Yeah,
absolutely. And Dimple, so when you were finding this was really beautiful. I mean, you're finding of like enhancer specific point alteration, point mutation from T to A to T allele, and the significant effect that what we see. So this was, I believe, a somatic alteration, right? And my question was, what was the frequency of recurrence of this specific mutation that you found in the enhancer?
So, I mean, this was germline mutation.
Oh.
Yeah. And it is a rare mutation. We only see in about 6% of the population. But the 6% of population had much more aggressive prostate tumor as opposed to the other prostate tumors.
Oh, so that was more associated with the haplotype and I mean the ethnicity that what you kind of like mentioned about. Have you also looked at the Indian haplotype?
So this is where, I think the future work should be concentrated on. Of course, there's a whole genome sequencing effort is going on in Indian case scenario, but I still think we are so slow. And, I'm sure once we have this whole genome sequencing data in normal and, tumors, I think we'll be able to identify not only the common, but the rare events also.
So recently, Indigen has, I mean, they have already published, it was published in NAR. I'm sure you must be aware of like 1,000 healthy samples. And the data, I mean, not the raw ones is available, but at least a certain level of data is available publicly. Maybe that could be a good place to get started with that, even though that's an healthy individual, to see the frequency of these alterations.
Exactly. So, Amit, since enhancers, like Sabri was saying, identifying enhancer is highly tissue type specific. So, for example, an enhancer which is present in cancer cell, the liver cell may not be there in heart cell. So, this is where we have to have focused efforts, For example, if I want to see mutations that are associated with heart function, then I can align heart specific enhancer and see if those are mutated.
Okay.
This is unlike the coding genome because coding.
Right, but then you're addressing more in the somatic space, right? I mean, you're talking about germline. If you're taking in healthy individual, it should be that pan, it should be there in all the tissues.
This is why I think many samples are needed to capture all that.
Absolutely. I mean, I'll be very happy to talk more about it. Thank you both of you on behalf of the organizers. I mean, I'm not the person to kind of like doing that, but I'm really happy that you could come to this clinician specific meeting, and there is a lot more that both to like learn from each other.
Thank you.
Likewise, a lot of learning. Thank you, Amit.
Thanks, Abhi. Thank you. Sorry, Pratik, I took a bit of your time. Please go ahead.
No, sure, sir. Those were nice questions and very nicely answered by both the faculties. So now, because we are running out of time, the next molecular tumor board is requested to be concluded in 15 minutes rather than the pre-allotted time. So, Dr. Smruti, requesting you to kindly proceed with the molecular tumor board.
I'm not able to share